I was walking through the grocery store the other day looking at the yogurt aisle, and it just, it struck me how we've been trained to think about bacteria.
Oh, yeah.
It's so binary. You have the good guys, the probiotics, right? The gut flora that help you digest your lunch.
And then you have the bad guys.
Exactly. The infections, strep throat, all the things we just nuke with antibiotics.
It's a very comforting worldview, isn't it? It's clean, simple. We like to know who the villain is.
We do.
But as we talk about so often, biology just kind of laughs at our simple categories.
It really does. And today's deep dive is going to take that binary, crumple it up, and just toss it right out the window. We are looking at a paper published in Cell, I think 2026, by Yao and colleagues that, frankly, it kept me up last night. It suggests that in cancer, specifically breast cancer, a bacterium isn't good or bad because of who it is.
Right.
It's good or bad based on its zip code.
That is the perfect way to frame it. The study is called Divergent Tumor Immunity Determined by Bacteria Cancer Cell Engagement.
It's a mouthful.
It is a mouthful, but the core finding is it's startlingly elegant. They found the exact same strain of bacteria can be a deadly accomplice that helps cancer spread or a helpful soldier fighting the tumor.
And the only difference
is whether that bacterium is sitting inside the cancer cell or just floating outside of it.
Just think about that for a second. The same bug, two totally opposite outcomes based on a few microns.
Yeah.
So our mission today is to rigorously unpack this. We're going mechanism by mechanism. We have to understand the how a bacterium inside a cell hijacks the entire immune system.
And then the so what? Because the clinical data in this paper on cancer recurrence, it's just staggering.
It really is.
So we have a lot of ground to cover. We'll start with this paradox of location, then the molecular Trojan horse, the cGAS-STING pathway.
That's our main character today.
It is. And finally, what this all means for actual human patients.
Okay, so let's start with the context. We've known for a while that tumors have bacteria in them, right? This isn't brand new.
Right, the tumor microbiome. For a long time, scientists thought it was just contamination, or opportunistic infections.
Sure.
But recently, we've realized they're actual residents. And previous work showed that intracellular bacteria, the ones hiding inside the cancer cells, they act like a structural crutch.
Like a little exoskeleton.
Oh, yeah.
Endoskeleton, I guess.
Exactly. Think of the cancer cell in the bloodstream like a raft in whitewater rapids. The bacteria inside just reinforces it so it doesn't break apart.
Okay.
But this new paper by Yao and colleagues asks a much bigger question. They weren't focused on the structure. They wanted to know, is the bacteria talking to the immune system?
So to test this, they set up what I think is just a brilliant experiment. They used the PyMT mouse model.
A classic model.
It is. The mice spontaneously get breast cancer, and it eventually spreads and metastasizes to the lungs. And they infected these tumors with Staphylococcus xylosus.
Which is a very common bug in these mouse tumors. It's not some exotic pathogen they picked.
Okay, but here's the genius part. They needed to separate the effects of the bacteria inside the cell from the ones outside. And you can't. You can't exactly go in with microscopic tweezers. So how did they do it?
They used the physical properties of antibiotics. This is such a cool point. They used two different regimens.
Okay.
Regimen A, penicillin, and streptomycin. These are big, bulky molecules. They're great at killing bacteria floating around outside the cells, but they literally cannot get through the cell membrane. They just bounce off.
So if you use penicillin, you're basically stripping away all the external bacteria, but the ones hiding inside the cancer cells, they're safe.
They're safe and sound. Precisely. Then you have regimen B, doxycycline.
Dox.
It's lipophilic. It loves fat. So it can just slip right through the cell's lipid membrane like a ghost.
And it kills the ones hiding inside.
It kills the intracellular bacteria.
So you've got one group of mice where only the inside bacteria survive, and another where the inside bacteria are dead. What happened?
The difference was, it was night and day. They looked at lung metastasis. In the mice where the intracellular bacteria were alive, and that's the penicillin group, metastasis was rampant, cancer everywhere.
Wow.
But in the mice treated with doxycycline, where they killed those inside bacteria, metastasis it plummeted.
I want to put a pin in the numbers here because they are just wild. In a post-surgery model they ran, the recurrence rate went from 65% or down to 6.7%. Just from killing those inside bacteria. That's not a subtle effect. That's That's practically a cure.
It's massive, but here's the skeptical question you should be asking right now.
Is the antibiotic just killing the cancer?
Exactly. Or is the bacteria just, I don't know, a little battery pack for the tumor? Is it feeding it?
Right, yeah.
So to rule that out, they repeated the whole experiment in NPSG mice.
And these are mice that are severely immunocompromised.
Severely. No T cells, no B cells, no natural killer cells. They basically have no adaptive immune system.
So what happened then?
The doxycycline treatment stopped working.
What?
Killing the intracellular bacteria did nothing to stop metastasis in those mice.
Okay, wait. Let's freeze frame that because the gravity of that is huge. If the mouse has no immune system, it doesn't matter if the bacteria is inside or outside, the cancer just spreads anyway.
Correct.
Which proves the bacteria isn't helping the cancer directly, it's manipulating the immune system to help the cancer.
It's weaponizing the host's own immune system against itself.
It's not the engine, it's the driver steering the car.
That's the perfect analogy.
Okay, so before we get into the molecular detective work of how it does that, let's just do a quick recap.
Good idea.
1, tumors are not sterile, they have bacteria. 2, location is everything. Inside the cell drives metastasis, outside does not.
And 3, and this is the key, this whole process depends entirely on the immune system. The bacteria is hacking our defenses.
So now the question becomes, who is being hacked?
Exactly. The immune system has so many players. T cells, macrophages, who's the traitor?
The researchers looked at the lungs of these mice, the site of metastasis, and they just saw a flood of one specific cell type, the neutrophil.
Neutrophils are so fascinating. They're the grunt soldiers of the immune system, you know. They're usually the first responders to an infection. They show up, they eat bugs, they release toxins, they die.
But here they weren't acting like soldiers.
No, they were acting like bodyguards for the tumor.
So the team used single-cell RNA sequencing, which is, for anyone who isn't familiar, it's like taking a census of a city, but instead of just counting people, you interview every single one to see what they're thinking and feeling.
A perfect description. And this census revealed a split, a fork in the road.
Okay.
When the bacteria was inside the cancer cell, the neutrophils turn into what the paper calls the G2 cluster. These are suppressive neutrophils. They express really high levels of a protein called PD-L1.
PD-L1, that's the famous checkpoint, right? The don't eat me signal.
Yes. PD-L1 is basically a fake ID that says, I'm a friendly, don't shoot. When these neutrophils get coated in PD-L1, they bump into the T cells, the specialized assassins, and they just shut them down.
They paralyze the immune response.
Completely. So the bacteria inside the cancer cell tells the neutrophil to put up a do not disturb sign and block the T cells.
And the flip side.
The flip side, when the bacteria was outside the cell, the neutrophils became the G4 cluster. And these were antigen-presenting neutrophils. They were hyperactive. They were alerting the T cells saying, hey, threat over here, attack.
This is the mind-blowing part. The same bacteria. If it's outside, the neutrophil sees it and says attack. If it's inside, the neutrophil gets brainwashed into saying protect.
So now for the million-dollar question.
How does the cancer cell know the bacteria is inside, and how on earth does it tell the neutrophil? Because the bacteria is inside the cancer cell, but the neutrophil is the one changing its behavior. There must be a signal.
There has to be a signal. And this is where we get into the deep dive part, this mechanism. It's like a Rube Goldberg machine of cellular signaling. It all starts with a break-in.
Okay.
When xylosus invades the cancer cell, it's not a gentle process. It physically disturbs the cell's internal skeleton, the cytoskeleton. And this stress causes something very dangerous to happen.
Which is?
Double-stranded DNA, dsDNA, it leaks into the cytosol.
The cytosol is the liquid goo inside the cell. And DNA is supposed to be locked away in the nucleus, right, in the vault.
Exactly. Finding DNA floating in the cytosol is like finding furniture on your front lawn. It's a massive danger signal. The cell knows there's been a break-in.
And it has a sensor for that.
It has a sensor specifically for this misplaced DNA. It's called cGAS.
cGAS.
Cyclic GMP-AMP synthase. When cGAS finds this loose DNA, it binds to it, and it triggers a pathway called STING.
STING. Stimulator of interferon genes. I know STING, we talk about it in immunotherapy all the time, but usually we want to activate this STING, don't we? It tells the body, we have a virus, send help.
Yeah. Usually, yes. And this is the tragic irony of the whole story. Normally, STING triggers interferons, which are these antiviral warning flares. But here, in this specific context, a tumor cell invaded by bacteria, the wiring gets crossed.
So what happens?
The cancer cell doesn't make interferons. Instead, this specific S-STING activation triggers the production of a totally different protein, It's called IL-17B.
IL-17B. Okay, I know IL-17. That's an inflammatory thing usually made by immune cells.
Usually by T cells. But IL-17B is different. It's an epithelial-specific ligand.
Meaning the cancer cell itself is making it.
The cancer cell is making it. It pumps out this IL-17B, and it acts like a long-range missile. It travels out of the cancer cell, and it hits receptors on the neutrophils.
And it tells them.
It reprograms them. It tells them, don't become a soldier, become a suppressor. It drives them into that G2 state we talked about.
Okay. I want to trace this chain reaction one more time because it's so critical. Let me see if I have this right.
Go for it.
Step 1. Bacteria invades the cancer cell. Step 2. That invasion breaks things, so DNA leaks into the main room.
Yep.
Step 3. The sensor, cGAS, sees the DNA and pulls this S-Bing alarm.
Right.
Step 4. But instead of calling the police, which would be interferons, the alarm is rewired to call the mob. That's IL-17B.
Huh. That's a fantastic summary, yes.
And then step 5, IL-17B goes out and it bribes the neutrophils to protect the tumor.
That's it. It's a Trojan horse that rewires the building's own security system. The bacteria is the horse, but the cGAS-STING pathway is the gatekeeper that gets tricked.
And the researchers, they proved this right. This wasn't just a theory.
Yeah, they went hard on the verification. They used something called a transwell assay. It's basically a petri dish with a filter in the middle. They put bacteria on top and the cancer cells on the bottom.
So they were close, but they couldn't physically touch or invade.
Exactly. No invasion.
And the result?
No invasion means no cytoskeleton break, no DNA leak, no IL-17B. The process never even starts.
And they did genetic knockouts.
Right. They just deleted the gene for cGAS or for S in the cancer cells. And even if the bacteria got in, if the alarm system itself was cut, no IL-17B, no metastasis.
Wow. And the final nail in the coffin. They just added pure IL-17B directly to neutrophils.
And boom, those neutrophils instantly turned into traitors. It's a completely watertight mechanism.
It's terrifyingly efficient. But let's just briefly flip the coin. Why does the extracellular bacteria, the one floating outside, make the neutrophils good?
It triggers a completely different pathway. Bacteria on the outside, they interact with surface receptors called toll-like receptors, or TLRs.
And these are the standard I see a germ receptors.
Exactly. And when TLRs see the bacteria, they trigger NF-κB, which is your classic inflammation signal. But, and this is critical because the bacteria never enters the cell, there's no DNA leak, cGAS and STING never turn on.
So you get the correct inflammation signal without the corrupt IL-17B signal getting mixed in.
Precisely. The neutrophils hear the real alarm and become the good guys, the G4 antigen-presenting cells that help kill the tumor.
It all comes down to that one invasion step. But, and here is the big but, I know you listening are screaming right now, we are not mice.
We are not mice.
Does this happen in human breast cancer patients?
And this is where the paper becomes so clinically vital. The researchers analyze human breast cancer samples. Now, xylosus is a mouse bug. In humans, the dominant bacteria they found was Staphylococcus epidermidis.
Which is... it's everywhere. It's on my skin right now.
It is. But when it gets inside a breast tumor, it acts just like the mouse version. They infected human breast cancer cell lines, MDA, MB231 with S. epidermidis. And guess what?
Let me guess. DNA leak, cGAS, STING, IL-17B.
The exact same pathway. It's conserved through evolution.
And they looked at actual patient data.
They did. They went to the Metabric database, which is this huge collection of breast cancer genomic data, and they created what they called a bacterial invasion signature.
So looking for the genetic fingerprints of this pathway.
Right. And patients who had a high invasion signature, they had significantly worse survival rates.
So if you have this pathway active in your tumor, you are much more likely to die from your cancer.
Yes. And their tumors were just full of those traitor neutrophils, the GMDSCs, which is the human equivalent of that G2 cluster.
This brings us back to the recurrence model I mentioned at the start, because for a patient, the scariest thing isn't always the first tumor. It's the cancer coming back years later.
Exactly. So the authors simulated this. They grew tumors in mice, let them get established, and then they surgically removed them, just like a mastectomy. At that point, the mice looked cured.
But we know that cancer cells often escape before surgery, micrometastases.
Right. And in the control group, where those intracellular bacteria were left alone, 65% of the mice relapsed. The cancer just exploded in their lungs.
65%, it's a coin toss.
But in the group they treated with doxycycline, that antibiotic that gets inside the cells, the recurrence rate dropped to 6.7%.
From 65 to 6.7, that's the difference between a tragedy and a success story.
It really is. It suggests those dormant cancer cells floating around your body. They're being kept alive, and they're being protected by the bacteria hiding inside them.
And if you kill the bacteria, you strip away their shield.
And the immune system can finally see them and clean them up.
This changes. I mean, this could change everything about how we approach treatment after surgery.
Yeah.
Let's bring this home. The implications here are just huge.
Well, the biggest one is about antibiotics. There's been this long debate in oncology. Generally, we try to avoid antibiotics during immunotherapy because you need a healthy gut microbiome for those drugs to work well.
Right. We've told patients, don't take antibiotics if you can avoid it, because if you wipe out the gut, the immune system gets weak.
But this paper introduces such a crucial nuance. It says, yes, protect the gut, but if you have intracellular bacteria in your tumor, you absolutely must kill them.
So it's not antibiotics, yes or no. It's which antibiotic and where.
Exactly. It has to be a precision approach. Maybe we give doxycycline to kill the tumor bacteria, but we pair it with, say, a fecal transplant or targeted probiotics to protect the gut.
It also makes me think about pathology. You know, right now a pathologist looks at a slide and they say it's stage 2, it's estrogen receptor positive. They're not reporting on the bacteria.
They're not even looking for it. But I think bacterial mapping could become standard. Imagine a report that says HER2 positive, Staphylococcus positive. That second piece of information could be just as critical for choosing your treatment.
And what about STING? You mentioned we have drugs that are trying to activate STING.
This is the danger zone. We have spent billions developing STING agonists to try and wake up the immune system. But this paper shows that if a tumor has intracellular bacteria, activating STING might be the worst thing you could possibly do.
You'd be pressing the button that calls the mob.
You'd be supercharging the production of IL-17B and creating more of those suppressor neutrophils. It just proves that in biology, context is king. A pathway that is good in a virus infection can be bad in a bacterial tumor invasion.
It's a really humbling reminder that we aren't just individuals. We're ecosystems. We're walking around with trillions of bacteria, and in the case of cancer, some of those passengers are grabbing the steering wheel.
A very high-stakes negotiation is happening inside those cells.
Well, this deep dive has certainly changed how I look at the good versus bad binary. It turns out it really is all about the zip code.
Location, location, location.
This deep dive was based on the paper Divergent Tumor Immunity Determined by Bacteria Cancer Cell Engagement by Yao et al., published in Cell in 2026. A huge thank you to the authors for this truly groundbreaking work.
It's a paper that's going to be cited for years to come, I think.
And a huge thank you to you, our listener, for sticking with us through all the molecular weeds. So I'll leave you with a question to think about. If bacteria hiding inside tumor cells are manipulating our immune system, should standard cancer pathology start to include bacterial mapping?
Keep connecting the dots.
We'll see you on the next deep dive.